meta-analysis for binary outcome in SPSS to draw a forest plot and funnel plot. So what is a binary outcome? A binary outcome is a variable that have two levels in it. So it is a nominal variable, a categorical variable that have only two levels in it, yes or no, dead or alive, agree or disagree, have the disease, doesn't have the disease. And the risk ratio is the risk of the probability of an event occurring in the treatment or intervention or exposure group compared to the probability of the event occurring in the control or reference or unexposed group. So what is meta-analysis? Meta-analysis is a statistical method that is used or performed to combine data or results from several independent research studies. The objective of the meta-analysis is to generate and calculate the overall estimate of the effect size for specific intervention or exposure, in this case the risk ratio, by pooling all the results from these multiple studies that investigate the same research medical question. So the objective of meta-analysis when performing meta-analysis for a binary outcome and the effect size is the risk ratio, the objective is to compare two groups, the control group and the treatment group, and look for the risk ratio for both groups and determine an overall risk ratio for all the studies included in the meta-analysis. So the meta-analysis also will measure the variability among these results from these research studies by performing a heterogeneity test, which will examine and test and determine the degree of inconsistency or variability in the result that are extracted from multiple studies. And also the meta-analysis will generate a value for the heterogeneity test, a statistical value, and an I-squared value, which is expressed in percentage. I-squared value of 0% indicates that there is no heterogeneity among the results, and an I-squared of 100% indicates that there is a maximum heterogeneity among or between the result extracted from several independent research studies. The meta-analysis outcome will generate, or output will generate, a forest plot and a funnel plot. So what is a forest plot? A forest plot is a graph or a diagram that displays the result showing the summary of the meta-analysis output. It does produce and generate a summary of the effect size estimate for each individual studies and the pooled effect size, and also produce a 95% confidence interval and a p-value. The forest plot consists of three sections or part. The first part is the descriptive statistics that describe each individual studies, the generated p-value, the 95% confidence interval, the risk ratio, the number of participants in the treatment group and in the control group, and also percentage weight or weight percentage, which reflects the influence of the size of the samples on the overall meta-analysis. The second section is a diagrammatic representation of the descriptive results, which is, or descriptive statistics, which includes the estimate effect size, the risk ratio, the 95% confidence interval. The third part is the overall part that produce an overall or pooled estimate effect size, in this case risk ratio, and a p-value for significance, and also a heterogeneity test outcome or output with an I-squared and a p-value. And also the meta-analysis will produce or generate a funnel plot. So what is a funnel plot? A funnel plot is a graph or a chart or a diagram that displays the effect size estimate for each individual study, in this case the risk ratio, plotted on the x-axis, and on the y-axis the sample size or standard error is plotted. The objective of generating a funnel or drawing a funnel plot is to assess the publication bias. Here in this data view in SPSS, I had inserted data for 12 studies, as in the first column or the first variable is study ID from study number 1 to study number 12. And in each study, the publisher has assigned participant or arranged the participant into two groups. A treatment group and a second group, which is a control group. So here we have the data for the intervention and the data for the reference group. And in the intervention group, we have here the T stands for treatment, and event happening, and then a number of event not happening, the total participant for the treatment group, and for the control group the number of events happening in the control, number of event not happening or occurring in the control, and the total number of participants. So here two segregated group, and the number of event in each group is determined with the number of no event. So here to compare and combine and look at the results from these 12 studies, and meta-analysis will be performed. And since the outcome from each group or from the study is in a format of a binary outcome, which have two levels in it: event happening, no event happening. So here the outcome is binary, and to perform meta-analysis for this binomial or binary outcome, then the risk ratio will be taken or used as an effect size estimate. To do the meta-analysis in SPSS, click on the Analyze option here, and then select the meta-analysis option here. And since the outcome is binary, then, and the data are raw data, not a pre-calculated effect size, then select the raw data option, and a new window will appear, which is the meta-analysis for binary outcome. And as shown here, there are two sections: treatment group, control group, and then study ID. Move the study ID to the variable for the study ID into this section. And for the treatment group, we have three variables. What we want here is the success, which is the event occurring, and the failure, that no event occurring. That is for the treatment group. And for the control group, for the success, then in the control group, move the event to this section. And for the failure, no event in the control group. So here we have the data sectioned into two parts: treatment group, control group, with event occurring, with event not occurring for both group. In the right hand section, the effect size measured here will be the log risk ratio. We could go for risk differences, but we'll stick with the log risk ratio. And for the model to choose, we'll stick with the random effect. If we click on criteria, no changes will be selected, or no options will be selected. For the analysis, this can be done when there will be a subgroup analysis, which will not be, in our case here, be done. Clicking in inferences, no major selection or changes. In the bias, no major or no more selections. In the time and fill, no selection. On the print section here is very important. We need the homogeneity or heterogeneity test. So test for homogeneity, heterogeneity measures, which is the I-squared, and effect size individual studies, and tick on the prediction interval under the random effect model, and then click on continue. For save, not going to save any. For the plot, which is in this case two important plot, the forest plot and the funnel plot. Though there are other plots, we'll stick with these two. So to select and generate a forest plot, tick on that box. For displaying columns, tick on the effect size, tick on the standard error, tick on the confidence interval limit, tick on the p-value, and tick on weight, percentage weight. And for variable, I'm going to move all the variable into the additional column by highlighting them all, and then click on the arrow. And in this section here, annotation, tick on homogeneity, tick on heterogeneity, tick on test. And for a reference line, tick on overall effect size and null effect size. Click on continue, click on OK, and output is generated showing that this meta-analysis has been performed, and a summary is generated. The data type is in raw, the outcome is binary, and the effect size measure is the log risk ratio, and the model is random effect, and the weight is inverse variance, and the estimated method is RML. Moving to this section, which is the effect size estimate. This is the pooled effect size estimate, which is the risk ratio or the overall risk ratio, with a value is generated here, and a p-value is less than 0.05, which is significant, and a confidence interval with a lower and upper limit, and the effect size fit within that limit. Scrolling down to the here, this section, which is the test of homogeneity. to look for heterogeneity or homogeneity among the result from several studies, the 12 studies. As shown here, the chi-square, which is Q statistic, is calculated and a p-value is given, and we need this p-value to be higher than 0.05 as shown here by the I-squared percentage, which is 21% heterogeneity. So there is low level of heterogeneity. So this table is the one for testing for heterogeneity between results from all the 12 studies. Moving to the next section, which is the forest plot. In fact, I'm going to go back again and select from the plot here, display the exponentiated form, and click on OK. Again, the plot is redrawn as shown here. The plot is consists or is made up of 3 sections: the descriptive statistics, the diagram, and the third part, which is the overall pooled effect size with the heterogeneity test. Let us look at the first section here, which is a descriptive. The first column shows the studies from study number 1 to 12, and the second label here is the risk ratio for each individual study is calculated, and a 95% confidence interval for lower and upper is given here, and a p-value for the significance of the association between the treatment to the control based on the estimate effect size, which is the risk ratio, is also given here. The weight and the weight percentage is given here. The scores or the number of events in each group and no event with the total for each study also is given here. So this is a descriptive statistical with some calculation for statistics for each study here. The next diagram or next part of this diagram is, as shown here, is just a reflection or a representation of the values for the effect size, which is the RR, the risk ratio, the 95% confidence interval. As shown here, these squares, blue squares, represent the effect size, which is the risk ratio for each individual separate study. Larger squares reflect the weight percentage, which means that larger studies will have larger squares, as shown here. The horizontal line here is the 95% confidence interval, and the red dotted or dashed vertical line here is the overall risk ratio or effect size. And this is also represented, which is here 2.34, represented by a diamond, and this horizontal line here, this one is the 95% confidence interval. So the overall risk ratio is the diamond here, which is 2.34. So this is the second section. Back again to the first section, as we see here, we get an overall risk ratio, which is 2.34, and a 95% confidence interval with the lower and upper limits, and a p-value of less than 0.05. As the p-value is less than 0.05 and it is significant. Looking at the last or the third section here, or part from the forest plot, heterogeneity test done here, and there are 2 types, tau squared, which is also given statistics for it, and the Q statistics, and we'll stick with the Q statistics and a p-value. So for the heterogeneity test, we need this p-value to be above 0.05, as I have explained previously, and that indicates low levels of heterogeneity. For the overall estimate effect size, which is the overall or pooled RR, a p-value is given, which is less than 0.05. The final part is to draw the funnel plot, and to do that is back again to the meta-analysis, click on the plot, and then tick on the funnel plot, tick in the funnel plot square here, and for labels move the study ID to here, click on continue, and then OK. Again, an output will be regenerated again with the forest plot and funnel plot. On the x-axis is the log risk ratio. In this case, it's not the risk ratio. And on the y-axis is the standard error. And the line on the middle here is the pooled effect size. And the blue circles the effect size or the risk ratio for each individual study. And from this funnel shaped diagram, it is possible to determine whether there is a publication bias. So the distribution of the estimate effect size will be equal above the pooled estimate effect size compared to the below one, below the effect size. So the distribution of the study RR should be equally on both section here, so it should be a symmetry. And if we count the studies here, 1 2 3 4 5 6 7 8 on the right hand side, and 1 2 and 3 on the left hand side, with 1 is outside the funnel. So this indicates there is a certain degree of publication bias, and this need to be confirmed by another test, which is the Eggers regression test. So this is the way to perform binary risk ratio meta-analysis for an outcome that is binary in SPSS, and produce a forest plot and a funnel plot.